It is 1 o'clock, so good afternoon, everyone, and welcome to the American Flora Endowment Grow Pro webinar series. My name is Lena Layton, and I am a member of AFE Young Professional Council. I recently graduated with a PhD in plant breeding, genomics, and genetics from the University of Georgia, and I'm currently an ornamental plant breeder at a Spring Meadow Nursery, Proven Winner Scholar Choice. I'm serving as the moderator for today's sessions on managing Impatiens Downy Mildew with variety selection and production practices. On behalf of the endowment, I'm excited to be part of AFE Grow Pro webinar series that features a new topic every month presented by an industry expert. The webinars are free to everyone thanks to the generous support of AFE sponsors. This session is sponsored by PanAmerican Seed and Syngenta Flowers. PanAmerican Seed is the world's leader, breeder, and producer of F1 and open pollinator flowers and seed. PanAmerican Seed markets its products to both wholesale and retailers and retail distributors around the globe. Syngenta Flowers is one of the largest wholesale breeders of hybrid flower and cuttings in the world, developing and producing flower seeds and cuttings for growers internationally. If you'd like to see all of our sponsors and their sites, or if you are a supplier and interested in becoming a sponsor for a topic, you can find that information on AFE website at endowment.org/growpro or on the link in the chat. Today's session has been pre-recorded in English. Margery Daughtrey will join us following the presentation to answer questions that you might have. We encourage you to submit your questions through the Q&A feature or the chat. We'll answer as many of our questions live as we can before the end of the hour. Any unanswered questions may be answered through a separate email exchange following this presentation. Also, this session is being recorded and will be shared to AFE YouTube channel. And through YouTube's accessibility features, you will be able to access closed captions and also other languages. Without further ado, I would like to present today's expert speaker, Margery Daughtrey. Margery Daughtrey is a senior extension associate with the section of plant pathology and plant microbiology of Cornell University. She has conducted a research and extension program on the management of diseases of ornamental plants since 1978 at Cornell's Long Island Horticultural Research and Extension Center in Riverhead, New York. She educates growers on management of greenhouses and nursery crop diseases, runs a diagnostic laboratory, and investigates controls for bloxwood blight, Impatiens Downy Mildew, and other terrible diseases. Daughtrey holds a Bachelor of Science degree in biology from the College of William and Mary and a Master of Science in plant pathology from the University of Massachusetts. She's a co-author of several books, including the recent compendium of Bedding Plant Diseases and Pests. Margery, welcome and thank you for presenting today on managing Impatiens Downy Mildew with variety selection and production practices.
Hi, this is Margery Daughtrey. Welcome to the Grow Pro webinar series. I'm going to be talking to you today about managing Impatiens Downy Mildew with variety selection and production practices. The sponsors for the session are PanAmerican Seed and Syngenta Flowers. So let's start out with what are the downy mildews? Downy mildews are diseases that are different from all the rest. The pathogen in this case is not a fungus, it's not a bacterium, it's not a nematode, it's not a virus, it's not a viroid. It's something called an oomycete, which is a pretty funny word. If you go back to the ancient Greek, you can learn that these were originally named this because they were thought of as egg fungi, with the word oon meaning egg, and the word mycetos means fungus. When you look at structures under the microscope that are found in the soil, they include this thing that looks like a fried egg, and that is actually an oospore of one of the oomycetes, particularly in this case, the downy mildew of Impatiens. And with time, that oospore, under certain conditions of moisture and nearness of a host, is going to germinate and put out a germ tube, as you see at the top there, and it will be able to then grow so that it can infect a new plant. The oospore is actually why we have a big problem with Impatiens. It's what allows the disease to linger in a particular location from 1 year to the next or from 1 month to the next. When you have disease in the landscape, you're going to see a progression from flowers being affected and falling from the plant, then leaves being affected, and then the plants themselves keeling over, as you see here in this image from Knoxville. All the organic matter contains oospores, and they're released into the soil, and they wait there until another opportunity to attack an impatiens plant. In Riverhead, we began having problems with this disease in 2011, in the fall of that year. And we saw very quickly that the problem would come back to the same place where it had been seen before. For example, in this stone planter, we knew there was infection in September of 2011. And then we went back the next June, and we could find downy mildew on plants planted into the same planter because people weren't yet aware that the disease would reoccur in the same patch of soil at the time. I had another neighbor who planted this cross of flowers each year. Annually, he had impatiens in his cross. Annually, he had Impatiens Downy Mildew in his cross after 2011. Now he plants begonias. It actually isn't as mysterious as you might think because you can see a little bit of this with your naked eye. If you look at the top there, that stem has little black flecks in it, and those little black flecks are actually the oospores themselves. If you cut those stems in cross section, you see some little pink spots, which are actually natural to the impatiens plant, but the little black spots are really these things, which you can see under the microscope, those egg-shaped structures again, that are the oospores of the downy mildew. These are the sexual spores of this organism. But even though downy mildews make oospores and they're really important and they're why we have a problem, that's not all that downy mildews do. They also make a second kind of spore structure. And here you see a sporangiophore with spores around it. They actually all fell off of the tips of those branches. Those are asexual spores that are produced very simply and quickly by the downy mildew. And in some cases, they can actually make another kind of spore called a zoospore after they land on a wet leaf, multiplying the disease by another 7 or 10 times. So your sporangiophores are actually what puts the downy into downy mildew. They're sometimes called conidia if they can't make those special little zoospores, but this is probably a familiar sight to you by now. Here's Rudbeckia with its downy mildew. Here's a pansy with downy mildew. Here's the bacopa with downy mildew. And here's the snapdragon downy mildew affecting very young plants. It can form a grayish coating on the snapdragon leaf or even a pale violet coating at times. And another familiar one is the downy mildew on coleus. When you see that sort of grayish, dirty look on the underside of the leaf, that is an accumulation of the sporangia on the underside of the leaf. The one that we're concerned with today is Plasmopara destructor, which is the impatiens downy mildew. And the type of sporulation that it does is shown here. It's a whitish coating that you'll see on the underside of the leaf. This disease was actually not new to us in recent years. It was recorded way back in the 1800s on the native impatiens, the jewel weeds, Impatiens fulva or capensis, and Impatiens pallida. And it just emerged in the United States within the industry in the year 2004. This is the range here of the Impatiens pallida, the yellow jewel weed at the top, and Impatiens capensis, the spotted jewel weed at the bottom. And those are the green areas, and you get a sense for how much of this plant there is and what a wide range it has. So back in 2011, we were surprised that our beautiful plantings in the landscape started sort of melting before our eyes. We'd never seen this happening in the landscape before, even though we'd actually seen a few little samples from greenhouses back in the year 2004. So here's a September 15th picture from 2011. Here's an October 11th picture from 2011. This is a disease that does not take prisoners. It does not behave in a gentlemanly way. You go from a full planting to a really ugly, devastated, a lack of planting. The landscaper evolution shown here is that the next year we came back and there were marigolds under this tree. Not a good site for marigolds, but they at least weren't getting the impatiens downy mildew and melting out. We saw many very ugly scenes in the landscape, and I'm including these just in case some of you are too young to have seen this. It was pretty extraordinary. We had impatiens looking like sea urchins. We had them just languishing in their window boxes. Here's a trial from Nora Catlin's annual field trial, where the flowers went and all the leaves went and everything just really looked like a lunar landscape. Things that were full blooming plantings were no longer that. And if you waited long enough, even the stems disappeared. There was a great deal of press about the disease in the year 2013, even the Wall Street Journal. And here's a list of some of the other places where articles appeared talking about Impatiens Downy Mildew. And believe me, this is not usual. Ordinarily, the press does not pay much attention to plant diseases. It made a huge impact on the gardening public. One of the first things we started working on were the applications of fungicides to try to solve this problem. And we did see some rays of hope in that. This is just one little slice of data to show you that heritage didn't work very well at all, but that we were getting very good results from an experimental phosphorus acid material there in the middle on the lower level, and also from Subdue Max at that time. We also started looking at the plants themselves, because a disease is an interaction of the plants with the pathogen. And quickly we learned that New Guinean patients were not susceptible to this problem. That was really a break. So people could plant the Impatiens hawkeri, which is a different species of impatiens, and not have to cope with Impatiens Downy Mildew. But if they were to grow double-flowered impatiens or butterfly impatiens, they would find that they were getting the disease. It was really common on anything that had Impatiens walleriana blood in it. So we began to look at other species of impatiens to see how widespread this problem was. And we were able to find that a number of other species of impatiens, some of which are slightly familiar and some of which are ones we never hear of, were susceptible to downy mildew. You'll see the Impatiens balsamina there, which is the balsam impatiens, one of the more familiar ones. And it's a good example of how, although the disease would affect these other species of impatiens, it never affected them in the same way that it affected the Impatiens walleriana, our very popular bedding plant. You would get patches of yellow on the leaves, and there was a whitish patch of sporulation on the underside, but there was not the defoliation and the collapse of plants that we were seeing with the Impatiens walleriana. Here's a picture of Impatiens capensis, the wild spotted touch-me-not. And we put that into some of our trials and looked to see what would happen. And we were able to get disease symptoms, but they were tiny little spots, nothing at all like what we were seeing in the trade with our bedding plant Impatiens. We could get sporulation on the underside of the leaf. We could prove that we had infection, but there was not a huge impact on the plant. Here you see an example of one of the other species. This is Flanagani with a patch of sporulation from downy mildew. Here you see Hochstetleri that did have some stem collapse from the downy mildew. Here's an Auricoma that was susceptible. Here's an Arguta that's susceptible. Anything that was walleriana or related to walleriana would collapse quite quickly, whereas these other species might show symptoms, might support sporulation, but they did not collapse in a really dramatic way. This is the kind of thing we were left with when we inoculated Impatiens walleriana and had it beside these other plants that showed just individual leaf spots. This was actually very good news because it told us that there were genes out there within the genus Impatiens that could be used to build ourselves better bedding plants. And that's what was eventually done. Here's a list to give you a sense of some of the different species that we looked at. If you look over at the left, those are ones where downy mildew was observed, but there's also a code there for severity. And at the top, you see very severe Impatiens walleriana. And then you'll notice there's a T after all of the others. That means there was a trace of infection. We looked hard, we found some. Technically, it's a host, but it was not being badly bothered by the disease. And then over at the right, you see a list of organisms, plants, where we saw the disease not occur whatsoever. We couldn't find sporulation of the downy mildew on these. That even included some that we knew in theory could be hosts, the capensis and the pallida we know are hosts, the omiana we know are hosts, but by and large, there were no sporulation. There was no sporulation showing on these plants, even under the kind of conditions that we supplied, which were very conducive to infection. We worked with James Keach and Mark Bridgen on some of these studies, and James categorized the Impatiens into two sources, listing the ones from Africa separately from the ones that were from South and Southeast Asia to see if, as a plant breeder, he could see a clue as to where we might want to go in the world to find more resistance. But the species that have asterisks here are ones that we did not find any sporulation on, and they occurred in both of those groups, the African and the South and Southeast Asian groups. There was a period of time then when the industry had a bad problem and set about working on it. And I think this is sort of an incubation period to let the plant breeders do their work. And then lo and behold, the fruits of the industry trying to combat the Impatiens Downy Mildew with plant breeding started coming out around 2018. And the first ones that I saw were the Imara XDR from the Syngenta company. And they came out with a line of different colors, which they had me test with code numbers, which are the numbers you see at the left. And these were examined to see how they would behave in an outdoor situation with inoculum of the Impatiens Downy Mildew supplied in abundance. And we really did give these a lot of opportunity to develop the disease. We had a 50% shade cloth overhead, and we irrigated them once at 6:30 in the morning and once in the afternoon to try to provide conditions that would really allow sporulation of the downy mildew and spread of the downy mildew. And perhaps you can make it at the right there, some little dusting of sporulation starting to show on that leaf. It's always much more of a bloom early in the morning, likes those wet conditions. We were extremely impressed with what we saw with the resistant Impatiens line that we tested. These plants were all inoculated or brought to the presence of inoculum about Memorial Day, and then they were allowed to grow under the same conditions and replicated trial for weeks. And here you see the way they looked on July 16th. The accent white and the super elfin white, which are older cultivars, were down to stems, whereas the new Imara white was still blooming and still looking very good. And here you see the same kind of thing in the orange, where we had a super elfin white here for contrast and nice blooming plants from the new resistant lines. This will give you some numbers to get a sense of that. Everything that's in blue here are the older types of Impatiens, whereas the yellow lines here are showing you the new white and new red that Syngenta had come up with. And if you look at the last column, you'll see the most contrast, because in comparison to an accent white that had 56 leaves per plant showing sporulation of downy mildew, and an accent premium white showing 71 leaves per plant with downy mildew sporulation, the white Imara was showing only a 7.6 leaf load of diseased leaves. And similarly, the red one showed only 15 leaves that were diseased, in contrast to 79 and 87 leaves per plant in the super elfin and the accent premium. We soon got to work with the beacon Impatiens as well, and they were also impressive. It was such a good time going from this era where all the Impatiens were melting out to an era where the Impatiens had been improved. And here you can see the beacon red up at the top. As of, this was an October rated trial. The plant looks terrific on October 16th, and the control plant at the same time had truly just melted away to where you could hardly even see any tissue in the pot. And here you see a nice fuchsia cultivar that behaved the same way. The beacons were performing very well, just as the Imaras had. This is a graph that will give you the same kind of information. The one bar there that's different from the others is the super elfin white, which is showing almost 55% infected leaves per plant. And at the same time, you have that entire class of cultivars, which were numbered. We don't have the names of those, but you can see that they're all showing very suppressed disease, 2 of them not even showing any sporulation. So this was a monumental achievement, I think, in plant breeding for the greenhouse industry to, in a short period of time, give us plants that could grow once again. The world was once again safe for Impatiens. This was the look of a group of those super elfin whites that were the controls, the susceptible controls in our trial. And in contrast here, the beacon salmon, for example, really impressive. And we're doing a trial right now looking at some white cultivars, and you can see the difference, the gradations, and how susceptible or how resistant a plant is. And a truly resistant plant is really a handsome thing. One of the things that we noticed last year in our trials was that the younger a plant was, the more susceptible it was to the disease. At the left here, you see an older plant that was inoculated with sporangia at the same time as the plant at the right, which was about a month younger. And in this particular slice of time, you can see there's sporulation all over that plant at the right, the younger plant, and there was no sporulation at all showing on the older plant. There's a connection between the age of impatiens and their disease susceptibility. This unfortunately means for you that as the custodian of the plants when they're young in the greenhouse, you have the responsibility for the most susceptible of the impatiens. You have the responsibility for the most susceptible of even the highly resistant impatiens. So take that and work with it so that you realize that you are needing to use protection in both cultural arrangements and also chemical choices so that these young plants don't fail while they're in your hands. The Plasmopara destructor is able to really create strong disease pressure on the plants it infects. And we need the plants to be highly resistant to it, but we're still only at the level of high resistance. And let's talk a little bit about what that high resistance means. This is a paper that I published with Jana Beckerman and William Davis and Karen Rain and Joanne Crouch last year to explain to the industry that the highly resistant impatiens cultivars that we know and love are not immune to the downy mildew disease. They are highly resistant, but they're not immune. And we did this by talking about the story of two California producers who experienced the disease within about the past year. The first one was a grower who had plants growing under shade cloth in a wet year, and he found that there was a great deal of loss of his crop, even on the resistant Imara impatiens. There was also a case in March of 2020 where plug trays were shipped from California to Washington, D.C., and they showed the symptoms of sporulation on the plants on arrival. This was added to by finding some problems with the beacon impatiens in Long Island greenhouses in 2020 as well. So this underlined to me the fact that we can trust these plants, but we can't trust them all the way we could if they were a marigold, for example. They have some susceptibility to the disease. It's just greatly reduced. We know that the cases that happened in 2020 were with normal Paranospora destructor. It was the standard kinds of strains that we've seen in other years. We know that we could take the downy mildew from those infected plants and inoculate both the Imara XDR rose and a beacon violet and see sporulation within 12 days. In many cases, when we're doing experiments, we'll see sporulation on inoculated plants as soon as 8 days. So even that time difference gives you an indication that these are highly resistant plants. They just aren't immune. Think about several things when you're considering the use of the resistant impatiens when you're growing them in the landscape. They're going to be performing beautifully because they do well under normal landscape conditions where you're not irrigating them twice a day and not giving them 50% shade cloth overhead. There are many things going on at the same time when you have a disease development. You remember the disease triangle with host pathogen and environment. The genetics of both the plant and the pathogen are important. The plant should be as resistant as possible. And the pathogen, we would like to hope, is not going to get any more virulent than it is already. But there are different strains of the pathogen, and there are going to be continually different plants available to us that will be more and more resistant to the disease. The weather, of course, is extremely important because the environment is so important to disease development. When we have rainier weather, that's going to increase the pressure, the disease pressure on an impatiens planting. It will even increase the disease pressure in your greenhouse by raising the humidity. When you're growing in a frost-free area, you have much more to work against because then the impatiens downy mildew might just blow in the vent. It could be out there growing on plants in the landscape nearby. The spores are very light, they can go a long ways. So if you're growing in a frost-free area, pay special attention to the chemical control that's needed in the greenhouse. When you go to do the chemical control, you'll have to be paying attention to resistance development, which might change at any time. At the moment, we know that mefenoxam is quite likely to no longer be highly effective against the Impatiens Downy Mildew. There have been serious problems in Florida and in other parts of the United States. We don't have that resistance here in our area in eastern New York right now, but it could come at any time. We also know that fluopicolide has shown some resistance, and also boscalid plus paraclostrobin has shown some resistance problems. So these are all chemistries that you need to use without putting complete faith in them. They can be part of a program, but don't make them the backbone of your program when you're trying to get disease management in the greenhouse. If there is inoculum already present in your greenhouse and you're trying to grow the highly resistant Impatiens, that's going to be a challenge for them. It's much better if you keep other Impatiens far, far away from the Imara XDR and the beacon Impatiens, because otherwise they're supplying inoculum and increasing the chance that those resistant plants can become infected. There are fungicides that work well against the disease. I've highlighted those in green at the bottom of this image. You can see the oxythiopiperine, Segovus, dimethomorph, which is Stature, manopropamid, which is Mycora, cyazofamid, which is Segway, and mancozeb, which is sold as Protect and Dithane and many other products. Those offer really strong tools to prevent losses of your young plants. Pay attention to frac groups when you're designing your program. You want to make sure that you don't try to rotate between Stature and Mycora, for example, because they come from the same frac group. Also, learn from previous years' experience and pay attention to the length of time that a particular material is able to give you protection. Some of the materials like a mancozeb might give you only 1 week of protection, whereas something like the dimethomorph or the oxythiopiperine are likely to give you more like 2 weeks. So all of this takes a lot of integration. And if you want to be a good Impatiens grower, hopefully you'll be able to put it all together and come up with a really strong program for producing the plants. Even if you're growing highly resistant Impatiens anywhere in the countryside, do use fungicides to protect them. There might be a difference in the number of applications in different parts of the country, but there is a need for fungicides in both situations, either growing in frost-free areas or areas that receive some frost. And while you're at it, remember that we always had other diseases and Impatiens too. It's going to be important to avoid conditions that would favor alternaria leaf spot or Pseudomonas bacterial spot. Both of those are really encouraged by wet leaf surfaces. So be a grower who understands the benefit of growing dry, at least with regard to the top of the plant. And also manage your thrips really carefully. If thrips take over your crop and are spreading INSV or TSWV, the Impatiens necrotic spot virus or the tomato spotted wilt virus, then you will lose your crop to another kind of enemy, which is airborne. So keep all of these things in mind to coordinate the production of your Impatiens so that they can be really healthy as they go out to the customers. My suggestions are to grow these highly resistant Beacon and Imara Impatiens. They're really wonderful, a huge breakthrough in the safety of Impatiens for us to grow now that we're in the era of this downy mildew. Don't mix them with other more susceptible types of Impatiens, and don't be fooled by things that sound like they're different. If they're Impatiens Walleriana, they are going to be susceptible to the downy mildew. If they are a hybrid of Impatiens Walleriana with other Impatiens, they're going to be susceptible. Use effective fungicides in rotation during production and pay a special attention to this if you're in the frost-free areas. I'd like to thank Nina Shishkoff, Alan Wyndham, and Rob Wick for helping me out by letting me use some of their images. And I certainly want to thank AFE for helping to get this message out to the growers, and also to thank the sponsors PanAmerican Seed and Syngenta Flowers for supporting the webinar series. I'll invite you to the next session as well. There will be one on sensors and advances in technology that will be happening on Tuesday, October 19th at 1 p.m. And reminding you also that to register for this and any other future or past recordings, you'll want to go to the website at endowment.org/growpro. And with that, I'll just suggest that you ask some questions, because I'd be happy to answer any other questions you might have about the downy mildew that affects Impatiens. Thank you.
Thank you so much, Margery, for your presentation. That was very informative. I would like to open up the session now for questions. So please submit them in the Q&A or just in the chat. And I will ask Margery if you have any questions. I would like to ask one question, waiting for the people to write down their questions. Why do we know why younger plants are more susceptible than the older ones?
No, I don't think we really do, but there are other plant disease systems where that's also the case. There is perhaps a maturity factor in developing a full-fledged immune system in a plant sense. So it may be that the plant's age helps it to have more physical barriers, let's say a thicker cuticle, and perhaps also some chemical barriers too. I think the main thing that is happening with the Impatiens Downy Mildew is that in some cases, the downy mildew is able to move very quickly systemically through the plant. And in other cases, the cultivar is such that it's blocked from moving easily. And perhaps there's something about the genetics of that, which is age associated. We don't know, but boy, it would be interesting to know.
Yeah, that would be very cool to know. So we've got a question in the Q&A. The question is, are you continuing to test new varieties in the Imara or Beacon series, and are they improving in resistance?
Interesting question. One thing that it makes me think of is that I really can't tell if they're better from year to year in some senses because the environment is different from year to year. So to do a really good comparison and get a really clear read on plant behavior, I need to have them all at the same time under the same set of conditions. So I might get an idea that they're getting better or getting worse. But at the same time, I don't think I'll really know unless I had several years of that kind of an experience. But yes, I am still doing some testing. I'm working with a group of people who have committed themselves to learning more about Impatiens Downy Mildew with support through APHIS. And I'm also working individually and helping Mark Bridgen with his work that is also dealing with looking for impatiens resistance. So that's something that's continuing here from year to year. Yes, indeed. There's a trial going right now, as a matter of fact.
Okay, thanks for your answer. I have another question. Do the you said that there is another species that was resistant, right?
Anything but the Impatiens hawkeye, New impatiens?
You said that one was resistant. Do we know why that is resistant? Is based on like basic genetics, or is some kind of physiological reason?
Good question. And the easy answer is always that it's something genetic. So that's always true. And since you're a plant breeder, I'm sure this gets too intriguing you, and I would love for you to do some work on it. Unfortunately, in the great scheme of diseases, the Impatiens Downy Mildew is, although technically on the books since the 1800s, it's sort of a new disease in terms of our modern interaction with it. And we don't get to the fancy kinds of understanding in the first 10 years or so of working with the disease. So someday I would hope that there will be graduate students at universities answering that very question. It would be really terrific to know. We were just very lucky that there was this pre-existing plant which performed beautifully in the industry, which looked something like an Impatiens walleriana that could be switched to by those who needed to avoid that disease. And I think they have different personality. I don't think that's a very complete replacement, but it certainly was great for us in those intervening years before we had the Beacons and the Imara XDRs.
Yeah, it's like an alternative, not like a straight replacement. Yeah.
Sure.
If you have any other question, please submit it in the chat or the Q&A from Margery. Use this time that she's here, we can answer live any of your questions if you have them.
People tend to be shy. They like to get you with emails afterwards instead, I know.
Yeah, please don't be shy. Submit your questions. Okay, I have one more. Can I still use the Subdue Max for control of downy mildew in patients?
That's a good question. Here in the Northeast, for example, we haven't seen a resistance to Subdue Max in our Impatiens Downy Mildew population that seems to have gotten started here years ago. In other parts of the country, most notably Florida, there's a terrific amount of Subdue Max resistance, mefenoxam resistance. So the answer is to some degree regional, but then there's also the fact that even if you're in an area where that resistance has occurred or is currently occurring, it's still useful to include that within a rotation. You just would want to be very careful not to rely solely upon mefenoxam because there's always a chance of resistance development in that particular chemistry. The way the label is written, you should use another material from a different frac group with it when you apply it. And that's because of awareness that it's something that downy mildews can become resistant to. So if you obey that label precaution, you're putting on another ingredient at the same time. You have the same kind of label suggestion with Adoran. So combining the two might be an appropriate choice. And I would really like to see it included within a rotation within an entire season of production because it is so terrifically effective when it does work. Very systemic, and it works beautifully against the disease if the pathogen has not become resistant to it. So I would say include it, but I would not use it as the last treatment going out the door when you're finishing your plants and sending them out to the landscape. And I probably would not use it at the beginning of the season if you've gotten a scare and you've heard there's some in the neighborhood. But work it in between other materials and always do the tank mixing as you're required to by the labeling.
Okay, thanks. We got another question in the Q&A session that it says, does Aliet still works for downy mildew?
Good question too. Aliet has effectiveness against this downy mildew, but it's not as strong an effect as the experimental phosphorus acid that we were testing at the same time. So I think it's got a contribution. I think it, again, could be in a rotation. I think that active ingredient is very effective against Impatiens Downy Mildew. I don't know that there's any resistance problem there. Perhaps we need to explore different formulations of it to get one that's got even more power than the Aliet does specifically. But there's efficacy there. So that would be a very logical thing to keep in your rotation. Yes.
Sorry, I was muted. Okay, thank you. We got another question in the chat that says, will packaging the plants moist affect the growth of downy mildew?
Okay, that's an interesting question too. And we deal with this kind of thinking when we're considering boxwood blight too, like things happen on the West Coast and then we get them on the East Coast. So what happens in between? One of the things that I notice, because I do a diagnostic lab, is that many things will happen in the box, okay? So if the plant is shipped in such a way that it's somewhat airtight and it's in transit for a couple of days, that's exactly the kind of circumstances that provides the high humidity that allows sporulation. So the sporangia come out from the stomates on the underside of the leaf under those high humidity conditions, anytime as soon as 8 days after the inoculum landed on the plant. So if the inoculum had landed 7 days before and you had 2 days of shipment, there could be sporulation showing on a plant that was innocent looking when it left the West Coast. So yes, there can be complications associated with the time of shipment and definitely humidity. So a wetter shipment probably would have more ugliness of downy mildew on arrival if indeed it happened to have been infected before it was shipped.
Okay, thank you so much. If there's no more questions, I would like to thank everybody for joining us today for another sessions of AFE Grow Pro webinar series. We encourage you to view our full calendar at endowment.org/growpro. Also, the link is in the chat. While you are there, please check out our past webinar recordings, other grower-related resources, and research reports available to you for free. Thanks to our industry support. Our next webinar will be on sensors and advances in technology, and it will be on October 19 at 1 p.m. Eastern. We ask you to please complete the brief survey about today's sessions, and because your feedback will help us continue to improve the monthly webinars and provide additional topics of importance. Thank you so much, everybody, for joining, and have a great day. Bye, everybody.